Okay, we have our panel all set up. Thank you. And we welcome aboard Dr. Elizabeth Bielski, Senior Pharmacologist from Office of Research and Standards within OGD, and also Dr. Zhen Xu, Senior Staff Fellow with the Office of Bioequivalence from OGD. So we are running a few minutes late, so let's see how many questions you can get in, and also allow you a break to go do the graphic survey. Okay, any questions in-house, please? Online.
We have an online question for Dr. Liangfeng Han. Does the agency suggest that it is acceptable for MDI to be non-Q1, Q2?
Yes, so as I mentioned in the talk, if it's not the Q1, Q2, we recommend that and the applicants take the option 2 that follows the traditional BE approach, including the clinical study. But we also understand that it's possible that if it's not Q1, Q2 the same, the applicants can also take the option 1, the alternate BE approach. The rationale for us is that to take the request that the Q1, Q2 the same in the option 1 is that we think is that the suspension-based MDIs are complex. It's likely that the non-Q1, Q2 formulation may cause the not the BE between the in vitro BE testing and the performance. So we recommended the Q1, Q2 the same in option 1 that reduce the likelihood of this possibility. But we definitely understand that it's possible that you develop non-Q1, Q2 formulation and you still see the BE between the test and the RD products. But in that case, we suggest you submit the pre-end meeting request to talk with us and justify your formulation. And that's my answer.
Thank you. Next question, please.
Another question for you, Dr. Han. Are there any concerns related to standardization of the charcoal methodology irrespective of the drug product?
Yes, not a concern. That we know that currently there is no standard charcoal block that method established. And we think that you can develop your own charcoal block the method, as long as that your charcoal block method can adequately block the GI absorption. And the dose of the charcoal block is not too high that can raise the safety concern. As long as that the safety and the efficacy of GI absorption blocking is satisfied, we think that the possible that the charcoal block method could be justified.
Thank you. We do have a question in-house, please.
Marie Ducharme, confirm at Université de Montréal. Just a comment about the charcoal block. I've seen many charcoal block studies where the charcoal block will deactivate the drugs going in the lung, because the charcoal block is given too close to the inhalation dose, and people do not, swish and swallow enough to remove the charcoal from the oropharyngeal area. So those studies are very tricky. But you can see it right away from the PK curve, because, in the PK curve, normally for those type of drugs, you see clearly 2 peaks. You see the peak from the lung, you see the peak from the GI tract. And when the charcoal block is not well given, the first peak is decreased. Okay?
Yes, we also see that problem, that just like you say, if the charcoal block is not properly administrated, it also blocks the lung inhalation. So there are some methods that can optimize. For example, that after you after you take the chocolate block, before the inhalation products was given, you can rinse your mouth, right? To swallow it, rinse your mouth, and that can reduce the lung absorption. Also, that you may consider use the maybe reduced amount, somehow reduce the amount of chocolate block, but I don't know if it's a timing, yes. 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 So the method, I think, definitely can be optimized. Thank you.
Thank you. Online questions, please.
We have a question for Dr. Ross Walenga. What options are available to applicants to discuss issues related to in silico modeling for orally inhaled drug products?
Yeah, of course, there's the pre-end of meeting pathway, which has been talked about at length in these workshops, but there's also the new MIE Model Integrated Evidence pilot program. So that offers a new way to talk about your model, because you can also cover multiple drug products in those meetings. So you could have a model that you're trying to use across your whole suite of products, and we can discuss it in a more kind of holistic manner.
Thank you. And we do have a webinar on MIE on our SBIA Learn repository. Okay, next online question, please.
All right, another question for you, Dr. Walenga. How can applicants develop regional lung deposition models when there are no in vivo imaging data available for the drug product of interest?
Yeah, thank you for the question. This can be an issue with some products. I've talked to some length about in vivo data, and there are some products out there where you've got literature results, but in some cases you don't. So I think if you've got that situation, of course, you could try to do the study yourself, but if that's not available to you, you could try a more, what we call a platform modeling approach, where you have several similar products that you do have data for, show that you have credible models for those products. And then as long as the product you're considering isn't too much different, it could still be used as a supportive model.
Thank you. Next online question, please.
This question is for Dr. Bielski. Does the agency consider alternative BE approaches for dry powder inhalers if the proposed generic product is Q1 or Q2, the same as the RLD with similar inhaler designs?
So you're asking if alternative approaches are available for DPI products that have similar formulations. We always consider alternative approaches for any of our guidances. And if you pay attention to we've been going through a lot of revisions for inhalation products. So if you've been paying attention in this arena, you'll see a lot of change going on. So I highly recommend you keep checking those periodically. And of course, if you have a DPI formulation that's Q1, Q2 or non-Q1, Q2, you can always come to alternative approaches. And you may see some of these option-based approaches that you see for the MDIs also being proposed for DPIs as well. I think those are, of course, always things that we will consider having meetings with you to discuss those options. And, you know, as anything, what we're going to include for those options are all going to be product specific. So it's a really good way to come talk to us, especially if you have something you think you'd like to propose to us. We're more than happy to provide our scientific input on your proposal. Thanks.
Thank you. Next question, please.
Next online question for Dr. Zhen Xu. Are there any differences between solution and suspension MDIs in the BE recommendations when using an alternative approach?
Thank you for the question. So for the recommendation for BE testing, I think for the solution and suspension product, they include the in vitro testing and PKB testing and CCEPR alternative study. They are similar, but from the drug product perspective, the suspension product, because it's a colored system, it's you can say it's a thermodynamically not as stable as the solution product, which is dispersed at the molecular level. So that could give you some more challenges or more studies that need to be done to do the B study. Thank you.
Thank you. Next online question, please.
Another question for Dr. Han. Will the agency accept a study where certain in vitro tests may not meet BE requirements, but the PK study shows bioequivalence?
Yes, it's possible that in the submission, we sometimes see that the applicant did not pass the solution studies, but it passed the PKB studies. And in some cases, that we will wait consider all the evidence, and sometimes we think that the PKB studies shows that the in vivo dissolution and adsorption, and in that case sometimes, but it really depends on case by case, and we accept that the submissions that fail some in vitro tests. Yeah, thanks.
I'd like just to add to that, as well as Ross pointed out, if you're failing some of your in vitro BE tests, it's a really good chance to use modeling as an option to kind of prove that it's not affecting the regional deposition or absorption. So again, even if you think you're failing and you have questions about that, please come talk to us. We'd definitely like to hear from you where your main challenges are coming from and which in vitro tests are really causing you the issues.
Thank you. Do we have more online questions?
Yes, we have a follow-up to the question regarding charcoal. Would you recommend after taking the charcoal, the subject should take some water to rinse the mouth out or not? And if water is used, will the charcoal be diluted and lost of the blockage?
Yes, actually, I think it's the good practice that you use water to rinse your mouth and swallow it before the drug administration. Of course, that you don't have to drink a lot of water to dilute the charcoal block efficiency. That is that means that you probably need to optimize your charcoal block method.
Okay, thank you. Next online question, please.
We have another question that came in addressed to Dr. Bielski. If Company A conducts an alternative BE approaches as per agreement with the agency, whilst Company B conducts a traditional BE studies, including a clinical endpoint study on the same generic product, will the agency consider Company B's ANDA more approvable?
Thanks for the question. I think there are multiple ways to show evidence of your local bioavailability. So I wouldn't consider one option better than the other option. I think if we have more than one option available, you can use the ones that are available. Even if we don't have those options and you have an alternative, we will also accept that. Again, there are some risks to take, right? As a company, if you're proposing something new, that might take a little bit more effort and more risk on your part because you're not sure exactly how the agency will take that. But if you discuss with us early on and you give the agency time to consider that, the more likelihood we may consider that early on. So I think it is a risk thing, right? Because if you're trying to do the clinical endpoint, we understand that's going to take time and effort and energy, but everyone knows how to do those. So again, I guess it does come down to a business decision, which way you'd like to go, but the agency considers every application as the data that it gets submitted to us. So we don't consider one more approvable than the other. It's all a matter of the evidence that you provide to us. And the more that you discuss with us, the more likelihood, you can have some success in whichever route you decide to take. Thanks.
Thank you. Another online, please.
Question for Dr. Zhen Xu. PSGs that recommend alternative studies in lieu of the CCEP study, it seems that the RA-PSD has been recommended in all ANDAs, but other studies such as dissolution, particle morphology, charcoal block PK/BE studies are selective and do not appear in all PSGs. Why is that?
Can you turn the mic on, please?
Sorry.
Thank you.
Yeah, thank you for a question. The realistic APSD is actually an advanced form of the conventional APSD that described in the USP compendium. So it's a more sensitive and reproducible and cost-effective method because it's in vitro test than the CCEP study. And the advantage of this is it provides a more clinical relevant condition compared to the compendium APSD. And then this highlights the APSD, the realistic APSD is a critical study in alternative study approach. So one of the important features that realistic APSD can avoid the actuation breathing coordination issue when it is tested with the actuation and the breathing has that synchronization. So so the realistic APSD, of course, it has its advantage, but also has its limitations. One of the things that we can foresee is the realistic APSD that when we test for example, the low specific resistance DPIs, it may exceed the flow rate of 100 liters per minute, which is the inertial sampling apparatus, the validation limit. So because the required peak respiratory flow rate, for a cascade impaction operation, and that's one of the reasons that the test is included in as one of the weight of evidence approach, but it's an important one. Based on like other alternative study, like the dissolution method, for example, it's also a pivotal study recommended in the PSG, but for those like the highly soluble and rapid dissolution profile drug, it may not be feasible for the dissolution testing. Yeah, thank you for the question.
Thank you. Dana, yeah.
Yeah, can I just add that? So aerosol performance for any inhalation product is applicable. So I think that's why you see realistic APSD testing in almost all the option one based approaches. It's applicable to almost anything because aerosol performance plays such a critical role, and clinical relevance is always key for us. But when it comes to other approaches, where we try to stick dissolution versus permeability, we really want to understand what's going on at the bioavailability of the site of action. If dissolution, your drug's slowly dissolving, it's going to be moving around, then the dissolution is probably the primary factor that's playing a role in that bioavailability. And that's not the case for every product. Sometimes you have something that's fastly dissolving and your permeability is going to be the thing that's dictating its absorption properties. So that in those cases, we don't necessarily think that the dissolution test is going to tell us anything that's as relevant. So that's where those morphology characterizations fall in that place too. Sometimes morphology plays a critical role, how it becomes bioavailable. Sometimes the morphology doesn't play as a big of a role. So I think that's where you can see the guidance getting more product specific, because we are trying to understand what's going to be the most critical product performance parameters that are going to affect that bioavailability. Thanks.
Thank you very much. And I think we've run out the clock on questions. So thank you very much for our panel.